around 1% right now. And we are practicing classical, and I can say I am at a 0.1% right now for classical. So, but after practicing 0.1% of classical, 1% of Shastra, and almost 70 to 80% of proprietary and patent medicine, I can still vouch for 0.1% that is classical. Because in those cases, if we are able to practice that 1.1% classical with the classical fundamentals and the classical good medicines available, the results are tremendous, which you can expect from sometimes the due respect to the proprietary and patent. And I'm not talking classical only medicines. Classical means the entire panch karma, upkarma and everything. So please have that mindset from this session that we are not saying this is wrong, we are not saying this is good. Everything should be there in some several forms. It's your mindset where you are evolved right now. It's not that easy as far as Ayurveda classical medicine practice is concerned. Thank you.
Yes, thank you very much, sir. As Somit sir already mentioned about hydroponics, the cultivation in water. So let's also hear from Ramesh Varier, sir, regarding the propagation.
Yeah, first a small clarification. I am not belonging to the only contemporary school. I am from the classical school, which is also into contemporary. Okay, so little bit of contemporary. We are all from that. So basically, we are 100% believers and practitioners of classical form. But because of the need that we I was attending a training program. The trainer said, "Impossible is a word that we use to describe something that we don't know." I think that is the first mindset that we need to have, especially as students. Whenever you think of something as impossible, please understand that as a mind telling you to take it easy, to remain in your comfort zone. Because if you say it is impossible, you don't have to try. The moment you say something you want to do and it looks impossible, think of it as an opportunity that you don't know something, so you have to learn. You have to go about it. There are several people in the world, like he was saying, who are already doing stuff. We just need to be open and ask questions and acknowledge our lack of knowledge. I think that will be a great starting point for us to acquire. We don't have to know everything ourselves. There are people who have done. We can always have discussions, collaborations, and get to know. I can acknowledge when I came out of Ayurveda college, I knew zero about modern science or modern medicine. It's all what I got through learning thereafter, just because I acknowledge that I don't know what is happening in several situations. So I think that is one message I would definitely want to give. And with respect to the growing plants, they have one more point to talk about that. I was part of one big conference, I don't remember which one, where I heard a very interesting perspective. They said that we are talking about things going, not being available and so that. So generally we say you should not harvest or you should not use natural stuff like that. A counter perspective was said, where he said, if you really have something that has got real commercial value, it will never go extinct. So if you want something not to become extinct, find out the optimal commercial value of that. People will ensure that it is cultivated. Today, if you cultivate tulsi, what money do you get? It is a waste cultivating tulsi as a main crop. Okay, there must be technology where you can do it along with others, where a farmer has nothing else to do, he can sow some tulsi and get it. At the market rate today, cultivating tulsi is not viable. Whereas you look at the high value plants, where they are in huge demand, I'm sure people will find a way of doing it. I think commerce influences us much more than we understand. So probably our exploiting the herbs in a manner that adds maximum value to at a global level will change the whole scenario. I think India will become a super supplier of the world. There is a huge potential here if we are able to turn the table and look at it that way. If you are still trying to keep Ayurveda as a low cost, which can only be purchased or used by a poor guy, I fear it is very deadly. Especially classical practice will find it very difficult to scale. Whereas large scale requirement is there, farmers are ready to cultivate, is the one message I wanted to give. Thank you.
During COVID time, actually, there is one research paper published about the guduchi and hepatotoxicity of the guduchi. The female patient consumed the guduchi, and there is a big article in all newspapers, and there is a research paper as well. So my question that being a practitioner or the student, how when other people as a patient or family member, they ask about these things, what should be our approach? Hepatotoxicity with the guduchi. The female patient during COVID consumed guduchi, and it caused hepatotoxicity.
Yeah, so I will tell based on our evidence. So this is the biggest problem is when we talk about safety, we talk about case studies. And when we talk about efficacy, we talk about RCTs. So, show me one RCT which proves that Guduchi or any heavy metal is toxic. Not even a single paper you will see in any. What it is just survey. That you found out on the US shops that certain product had high levels of heavy metal more than required limit. That's fine. But has there been a consistent toxicity study done in humans that when it was in that high content, it really did a... So then that way all our rasa aushadas are toxic because none of the rasa aushadas has heavy metals less than their toxicity. Now, so what I would like to tell is we have done 2 COVID studies, that too in diabetic, higher risk. They were in a toxemia, definitely. And we have used Guduchi for 60 days. The paper is under review. Now, that is another hypocrisy we have to face. That being from Ayurveda community, they will not allow even to publish you on servers now. Metric servers, we are stopped. Yes, high impact. We have been rejecting 8 high impact journals have rejected us. Why? Because now in that study, 60 days follow up, each patient got Guduchi. We didn't find any hepatotoxicity. We didn't find any nephrotoxicity.
Yes.
Not 1 or 2 patients. More than 50 patients were on. So again, the problem is we just make a kind of, a newspaper heading. But when we talk about science, let us see all of these individual case studies have enough confounders to prove that it may not be because of the drug.
We got this answer based on research from contemporary side. So now I would ask Dravagun expert Ajayan Sadanandan sir. So why these papers or why these people are actually able to speak like this on public platform? What is the truth or what is the reality?
So I think this is one of the good thing that someone we always criticizing, then we will start to think. So morning onwards, we are seeing and discussing so many things that how Ayurveda is understood. So you can see that there are 3, 6, 8 balsam plants are there, impatiens. All are the same species, same plant. But see the stem, see the flowers, everything is different. Like that in Guduchi also. There will be a Varna difference, there will be a Guna difference, there will be a Agrati difference. This is the fundamental thing we have to study. Expose yourself to physical matter understanding. Then you expose yourself to biological matter understanding. How we will understand? We will understand through the Gunas. Gunas is the basic thing we have to do. So if that study, look into that aspects, or those who person, I saw that paper and on that a photo was there. That is not Spora cordifolia, the Guduchi, what it is using. It is another species that is occurring on the coastal areas of Goa and Maharashtra. Another Tinospora species is used there. And they done the study on that species. We have another Tinospora is there, Tinospora malabarica, that is called as Kanda Guduchi. We have another Tinospora called as Tinospora crispa, that is having so many toppings. So all are different species. That's why our Acharyas are looked into the Panchbhaudika concept of a plant first. So all these are looking similar. It is color is different because its Mahabhuta is different. Its Agrati is different because its Mahabhuta is different. Like that we have to start to study. So we have to expose ourselves to the Panchbhaudika, physical understanding of the matters in the nature. From the basics, if you have such an experience, then surely you will get multiple inductions to observe the factors in the patients or in the kashayas or in the tablets, everything. Then it will give an insightful thoughts and linking of the things. This is lacking. This is starting thing. We have to do the application of the pratyaksham and anumana. So here my opinion or everybody has to look into this aspect is first the child has to be exposed to understand the nature, to experience the nature in a manner that without the knowledge, he has to enjoy fully and experience the nature. So coming to this Tinospora, if you see the leaf of that Tinospora, each leaf will be drying in each specific pattern. If you see this species of the tinospora, what used by that patient, it will dry off by shrinking like this. But if you are see our tinospora cordifolia, it will not be dry, shrink like that when it is dry. It will be always flat. So it shows that that much rukshatva is not existing. The snigdhatva is existing there. The same thing, if you are doing aquaponics and producing brahmi, and if you squeeze it, how much froth will be there? The akasha tattva will be reducing there. But if you are taking it in a pond and do squeeze it at 10 ml, the akasha tattva will be more than 3/4. So there is no need of that much of biological or chemical understandings. Our acharyas are not came to this level with these types of techniques. Surely we can bridge out. You can bridge out wherever it is necessary. There we have to put forth. So I have a humble request to all the students here is you are growing up, you open up your observation techniques by our acharyas concepts, then surely it will change into another level that you will become a scientist. Then together with you will become a Vaidya to a next level. So that's the ideal example of this tinospora cordifolia that has happened. And as Sara has told, it's completely a biased study in all aspects.
Thank you so much, sir.
Yeah,
so here you can see, contemporary side speaking with the research base, and here you can see the importance of Dravya Guna Vijnana, because Guduchi is our favorite, drug of choice when it come to exam or when it come to practice as well. But there are, like different species of Guduchis are as well. So we need to study about those things.
Yeah, just to add on one thing, sir mentioned that heavy metal aspect. So I just give you one example of our clinic. So we were giving Rash Shastra medicine to a patient for around 3 months, and one allopathic doctor raised an issue when she went with her husband for some other issue for her husband. So he asked that why you are not coming to us. So she said, I'm taking Ayurveda medicine and these are the medicine. So he said, boss, it will damage your liver and kidney. These are heavy metals. So they came to me that you are giving us heavy metals from last 3 months. Our allopathic doctor is saying that it will damage my liver and kidney. So I said, okay, ask them how it will damage your liver and kidney. So, and in fact, I said, okay, let me communicate. So I said, okay, if I will do a blood profile of all the heavy metals in this patient, and if all the range is in normal, will you accept that these medicines are safe? Because that is the only proof if some patient is taking from 3 months, and we did the blood profile and all the heavy metals were under the permissible limits. So that means if your blood is not having heavy metals as per the permissible limits above that, after taking 3 months of Rash Shastra, 3 years yogas, because due to some respiratory system issue, we have to give that. So that means these medicines are purified, processed in a nano form, in a Rash Shastra medicine, which will not damage your liver and kidney, period. So as a clinician, you have that tool always, because maybe you are not a researcher, you are a small clinician like me. So you have that tool of doing blood analysis of all heavy metals after confidently practicing Rash Shastra or any other medicines with good companies. It will not damage any any vital organs for any patient. Thank you.
Thank you very much, sir.
Can I add one more perspective
Yes,
to
please,
this?
sir.
I think the other approach also which we need to look at is the moment we hear something being published, we immediately become defensive. We immediately try to justify, oh, that is all wrong. See, first we need to go through it in detail. Then itself we'll understand the sort of biases that are involved. Most of our studies also are criticized in the name of bias. Most of our positive findings are pushed away in the name of bias. The same bias exists in most of these studies which are coming against also. So we just need to use the same tool that they used to kill us, use the same tool and go through that study in detail. We'll be able to find out the assumption that they have come through is not very well organized. So that is in fact what Somit was saying. Very often the study by which they came to that conclusion is very often, blowing up out of proportion. But that does not mean there cannot be a problem. At the same time, as scientists, we should be open-minded. We cannot say that Guduji will not harm the liver of anybody. Can we say that? Not Guduji, even Shira can harm somebody. Even food items can harm somebody. So let us be open. We cannot brush aside one study which is against an Ayurvedic product or herb and say it is all bogus, against Ayurveda by vested interest. This is how we start responding. And so people will write us off as another rhetoric. We should respond to it as scientists. Maybe it is possible, but how many people who took Guduji had this? What is the prevalence? How likely is somebody to have? So anything can be toxic to anybody, but it's not that it is a common occurrence. So we should probably develop a little bit of a more open approach to this whole thing is what I wanted to add. Thank you.
So we will come back to you, sir. So going back to that question about cultivation, like as Somit sir
I'll
was
be
rightly
more
talking-
interested to talk about the toxicity and of Tinospora. Okay.
Yes, sir. We were previously talking about the cultivation, the conservation of plants in terms of, as sir was mentioning about hydroponics and artificial intelligence. Being on the classical side, what are the conventional methods that have been followed, sir?
See, ideally, cultivation is not advised or advocated by the classic. So the particular classical word, afal Christ, it is there. So afal Christ means the medicine or the medicinal plant should be cultivated from the natural habitat. And if it is cultivated, it means you are removing the stress factor. And most of the medicinal plant having medicinal property due to secondary metabolites. And that's why this is the ideal thing, what I said it. But the requirement of the medicine, the amount of the medicine, we can try to follow to collect the drug from its natural habitat and to provide the cultivation, not exactly cultivation, but for survival of the medicinal plant in their natural habitat. This can be the only solution. Otherwise, if we cultivate any plant in anywhere, it is of no use. Say for example, Guglu is generally we found in the arid zone. And if we cultivate in the marshy land or in Anup, it will grow like anything. Lush green vegetative growth may be there, but the presence of Guglu will not be there. So this is the main problem. And it is, we can judge it easily in case of Guglu. But in most of the cases, this is a possibility. And that's why we have to try to collect the drug from its natural habitat and try to avoid the facilitation to the different herbs.
Yes, sir. One more question to you. In Dravyaguna Vijnana, we study so many actually herbs. But by law, what is actually Ayurveda drug?
Sorry?
By law, with Indian law.
Yes.
Yes.
By law,
What makes actual means to which herbs we can call
yes,
Ayurveda?
obvious. The Ukta Dravya. Ukta means where Rasagan, Varier, Vipak, and all these things are available in our classics. We should consider it Ayurveda. Because other than the Raspanchak or Leshokta or Anukta, we can go for it. We can, we can do research prospectively. But prospective research is having its own limitation, because if Ras, Gun, Virya, Vipak and all these things are available, then and then we can use it according to our need. Suppose the drug is act on dosha and dushya, multiple dushyas are there. By using different upakramas, a particular drug is having apatarpan, particular having santarpan. So full procedure, it is described in our classic. On that basis, we are using the drug described in our classics, and that's why it is called Ayurvedic. Otherwise, extra pharmacopoeial or Leshokt or Anukt, we can say it herbal, but can't say exactly Ayurvedic, because Rasgun, Virevipak and Karma are not exactly available in the classics. Thank you.
Thank you very much, sir. Before we go ahead with the next discussion, we have a question from the audience. So nowadays, many new companies come out with formulations having multiple formulation as ingredients. For example, for arthritis, all the following ingredients are there, like Maharasnadikwada, Magarindveja, Yogaraj Gugulu, Shalaki, Vacha, and Punanava, etc. How can we justify these new formulations?
Can you tell me Chavanprash has how many ganas? Chavanprash has how many ganas told in Ashtanga Hridaya. There is Trifala, there is Trikatu, there is Dashamulam. In Chavanprash, how many ganas are there? Many. So I think this is a wrong concept. See, Maharaja Prasadanyadi Thailam has more than 100 ingredients, many ganas and Shodhanadi ganas which you have ingrained. So I think this is a frivolous thought process which is propagated, that when you mix two kashayam, then I will think Vaidyanvatham will never, Chere Namudri will never practice. Have you seen his prescription, his combinations? So it is not that if you have proper gunakarma, as sir has talking about, if you have that gunakarma prayoga, you understand the samprapti, if you know the avastha, if you know to do chikitsa based on chanadhi vyadha avastha kaulo bhaishya jagkrit, and you make any combination based on existing, even proprietary medicine, if you know where to give, you are doing Sutta Chikitsa. So it is a misconception that even what you call as one formulation like Chavanprash is a combination of many ganas. So this, I think, should be left to the yukti of the Vaidyas. It should not be questioned. And all these can become a proprietary formulation. Can you say it is un-Ayurvedic? No.
Yes, sir. So with the continuation of this topic itself, the question to you only, sir, that like we have seen in one formulation there are n number of drugs, but many a times unavailability of the drugs or adulteration in the drugs. So abhav dravya or pratyanik dravya. So when you are manufacturing at large scale, what are the ideas behind that?
The first thing I will tell you with Guduchi controversy, we all use Amrita Churnam. Tell me, in original formulation in Amrita Churnam, it is told as Amrita Sattva. How many pharmacies are adding Amrita Sattva? I can challenge you. We all talk about the thing. Even my pharmacy is not adding Amrita Sattva. Or if they are adding, we are adding the totally adulterated form. So let us understand first this. Second thing is, there is no cap that I was just discussing before our interaction with sir, that our founder, Rama Varier ji, developed a unique Jatyadi formulation, which is very unique, using only 3 ingredients of API Jatyadi is in our Jatyadi Thailam. Even the Vaidyas who practice, even when I started doing research on that, I didn't know that is not the same API Jatyadi. When I looked at, it was totally, if you look at Varnadi Kashayam of AVP, what we make, the Bharlataka proportion is totally different to that of what is told in the text. Because of the Desha, he made the difference. Now, can you say it is a proprietary and it is not a Ayurvedic formulation? Now, in Adha Bhavav Pratindavyas, what we found in that when we took the Jatyadi formulation, which is told in API, and this formulation which was made by our founder, I was surprised. And this research was done in a European university. Himalaya has a head office there, you know, and in Europe, in Latvia. Now, what we found out, that the gene regulation from the patient, so it's also published now in one of the index journals, that we applied this Jatyadi both, and we took the fibroblast of the patient, and then we did relative gene expression of all the markers, inflammatory IL-6, TNF-alpha, all host of inflammatory cytokines, which are important for diabetic foot ulcer healing. And we were surprised that both formulations showed a similar healing. Now, the issue is, can we question the intelligence based on the guna karma which has been developed, which is very Desha specific in Kerala? Can we say, no, it is Abhav and Pratinidhi? No. I think Abhav Dravya and Pratinidhi Dravya cannot be adulterant. First, if there is a guna karma, I will be very happy. We should tell about Rasa Panchaka. In fact, I will again talk about a controversial issue of modern drug development, food technology uses sensory evaluation. I was talking to one of our colleagues here. They use taste, buds, smell, hearing, touch, smell, everything for evaluating the best food which is suitable, using a MRI, CT integrated with it to improve the, there is called as a wonder point, you know, where everything through your 5 sense organs that triggers the pressure point by MR, that is functional MRI they do. Now, can we start, see, again, we have to understand that we have to use these advances to even contemporize the understanding of five taste. Because rasa pradhana is like swada lakshana rasaha, and tongue is one of the best receptors. Now there is a lot of research done on e-tongue and all. So what I'm saying is, if what is known is limited, unknown is vast. So I think anukta aushadhas is hard or are very important time to use technology and other forces to give a rationality behind it. If it can help, we should be open to it. Thank you.
You see, the question that is, how do we justify? For your information, as Dr. Somit was talking about, Charaka in his Kalpasthana 12th chapter itself, he has said, as he rightly said, Pradesha Matra Betava Drashtavyam Shetchatam. So, based on the place that I have described, only 600 Virechara Dravyas, Swabudhyeva Sahasrani Kotirva Api Prakalpayet, Bahudravya Vikalpat Yoga Sankhya Navidyate. So, there is no limitation. So, as long as the safety and efficacy studies are established and any new combinations or whatever the combinations that Swabudhyeva, the Vaidyas do make it, they remain to be Ayurvedic drug by itself. And for your information, as you know very well, what is even Chavanprash, as he was talking about. If you look at the Bharata Vaishya Ratnakara, and there are varieties of Chavanprash. The ingredients are different, numbers by number. And as a matter of fact, including Dashamularishta, which is very popular, if you look at it, the 72 ingredients, it is humongous today in this situation to get all the 72 to make that. So just by the label of Dashamula printed on the label will not become Dashamularishtam, absolutely. So that is very important. Number two is that the preparations, that whatever that subsequent in the history of Ayurveda that these were developed. For Charaka, Chandra Prabhavati is a proprietary drug of Shangadra. And all these the preparations that which came out from Nagarjuna's the discipline is absolutely new to Charaka, Sushruta, Vagbhata. They are all actually proprietary of these people, Nagarjuna, Rasa Vagbhata, so many people. So every time as a new the times rolls on, based on the situation, that's what he says very, very clear. From that Pradesha, this is possible. From another Pradesha, you can think of something, Sobudhyerva, you can build up. So there is called as proprietary, which is not classical, and everything as long as these ingredients which are derived from Jangama, Udbida and Parthiva will be Ayurvedic.
Yes, so now we are taking a question from the students. Any one of you, especially the last benches. We have an expert from Dravyagun. Yes, sir, definitely will come to you, but I want one student. Because this is the platform, you can explore, what are the actually reality, and while practicing or manufacturing, what are challenges. By that time, we'll continue with our session, but I want one student with their questionnaire.
So earlier, as Ajayan sir had mentioned about cultivation of different species of the same plant across different geographical area to procure different varieties, different phytoconstituents. So at this point, would it be a compromise call, or what is the question of uniformity, efficacy, and potency? This is the question to contemporary first, and then we'll come to classical. So who wants to? Yes, over to Ramesh Varier, sir.
Yeah, I think it's again a question of the sensitivity scale that you're using. You can say, I will measure everything perfectly. One tomato is different from another tomato. Yes, the taste will be slightly different. If you want accuracy at that level of perfection, that I want exactly this tomato's taste, you will not be able to get many of those. But as long as you want a tomato, it will be available. So I think as medicine, we are not yet capable of actually, while theoretizing, while talking, we can say about such individualization and all that. I don't think any practicing physician has the capability to go that fine and say, I want this quality of guduchi to be given in so many milligrams for this patient. If somebody is practicing at that level of accuracy, I think the pharmaceutical Ayurveda is not suitable for them. Drugs manufactured by an industry can be sort of standardized to be within a range. That is at best what is possible. It is not possible to have precisely the same exact, the quantitative content of something. If somebody is claiming to be that, there is a lot of manipulation happening within that. I can give you an example of that also. I visited a very big pharmaceutical company in Indonesia where they were preparing standardized extracts. It was standardized to contain exactly a particular so much percentage of something. And I spoke to all those people. Because they were all from traditional medicinal background. I spoke to them, how is it that you can achieve? Because you are collecting from nature. How can you get the same level of consistency? Then they said, we have to manipulate. If the content is high, we add some more of the base material to make it like this. If it is low, we do a chemical process and remove certain other components from this so that the process is standardized. So I think if somebody is expecting perfect standards of, like a chemical drug, so many milligrams or so many picograms of this molecule is present in this drug, I think it is not suitable for natural product at all. Whereas you want to say that this particular herb is definitely present, there is a fingerprint, and that has to be quantitatively, it has to be not less than so much. At that level, I think is what standardization is possible. Even there, sometimes you don't get, the herb can come with slightly lesser, in which case you have to add more of that herb and balance it as long as you have standardized. I think that is what the expectation needs to be when we are talking about standards in Ayurveda. Thank you.
Yes, so this question is to Dr. Raveendranath, sir. Sir, you talked that when you consume the medicine in kashayam form, the matra is reduced. Like if it is 1 pulse, then it will be reduced to half pulse. So is there any research done on this, sir?
No. I think I could not do any direct research on that. I started giving it in that dosage, and clinically it is working very well because we were doing the other methods of preparation before. The same patients, when they come for the treatments, when we are giving in this reduced dosage, it is working in the same way. But that is I cannot say that it is a proper research that it is done. Clinically, our experience in that is working in that way, and it is working in the same. So, yeah, just one thing is, I was actually thinking about it, what is really the cause of it. Either the 10 ml kashayam or 15 ml kashayam which were being given normally by a practicing physician, we don't know whether the whole thing is getting absorbed or not. So either that one or the absorption of this material which is made like this in this way, I think the body is absorbing in a better way. I think either one of this should be the cause of it. This is what I assume.
Sir, in every kashayam, the dose is reduced in each
tablet. I can come out with a probable answer, though See, what I have found is there is better clinical experience with the tablet, mainly because of compliance. You give a bottle of kashayam, I have observed that also. We generally used to give the ready-made kashayam, 1 bottle was for 1 week. But I have hardly seen any patient who has completed a bottle in 1 week. They come after 10 days, 15 days saying quarter bottle is still left. Whereas you give it as tablet and you say the dosage is 6 tablets a day, invariably they are able to complete it. Maybe 1 dose was missing, but by and large they complete the 6 tablets, which itself gives you a much better result, is what I guess. So compliance drastically improves when you are giving it in a form that is more convenient to them, especially transportation and stuff like that. Otherwise, in today's world, people don't take especially difficult products, difficult to consume products unless they are extremely sick and there is no other avenue. Then they take it. Otherwise, they try to somehow manage. This is my practical experience, which probably explains why the lower dosage itself was clinically seemed to be significant.
Now we have a question from the audience side. Please, sir.
No, there is no question, but I just want to clarify on this Guduchi. See, earlier that particular case was raised around 6 months back, and that was CRISPR, as rightly mentioned, right? But later on, the same author, hepatologist from Apollo Hospital, Navi Mumbai, and one fellow from some other state, they together, both were hepatologists, and they said that we have now confirmed that the plant material is Tinospora cordifolia only, with the help of taxonomists, with the help of molecular biology, with the help of fingerprinting, everything they did, and they said that these patients died. Some of two patients were fatal, and then another were in ICU and all. And they gave interview to is no harm in saying because it's already in public domain, Times of India, right? And then there was a lot of chaos. Every now and then, all doctors were being, in Mumbai at least, people were asking, "Are you not giving Guduchi?" Right? And that was the time precisely the COVID was on the lower side. So everybody was eating Guduchi that time. Then we approached, being a association, myself, Dr. Nitin, Kochar, then another colleague of ours, we approached editor and we said, we would like to go through the details as someone said, and we need to read and find out. And we read that paper, we read those case studies, and we came to know that first thing, whether that particular patient was taking only Guduchi or anything else. And it was having other drugs also, other modern drugs, which were given for COVID, number one. Number second, they had other comorbidities. So how you can say it is only dying because of Guduchi? So then that editor was zapped. Oh, we never thought of like that. But the good part that we presented ourselves in a much organized way. We gave presentation, we put forth the data, and then we took promise from that editor. And henceforth, if you tend to get such kind of news, you better consult Ayurvedic group of experts. And then you decide, then you discuss if you don't mind, we don't mind to discuss with those two hepatologists. And then let us go ahead. But as anticipated, though they said that we will say sorry and all, which never took place. But at least message was through to them. So that is one part. And the second, not question, but just a curiosity. How many of this side, particularly Kashaya tablet and other things we were discussing, have we ever done PK studies, pharmacokinetic studies? Because if we do PK studies, then we can actually come to know the T-half, T-max and all that curves, and then it can be possible. Secondly, in Mumbai, there is one college of pharmacy where I am also associated, and Department of Pharmaceutics, professors from that, UDCT, and there is one more college, I remember. So we were coming together and developing methods, how to develop tablets. Because tablets can be done punching way, you can do with pellets, and there are many other ways. So which particular way, if we prepare which particular ingredients, and then how it can be more easily getting bioavailable. And I think that could be the ball game, right? Thank you.
Thank you very much, sir, for adding this point as well as the clarification.
Thank you, sir. So I have a comment to make here. Like we were discussing the difference between the compressed tablets and the kashaya, and there was a question regarding the studies. So there is one study under process in which we were trying to see the difference in the Shastrokta Ayush Kwat and the difference between the Ayush Kwat granules when given in a dissolved form to the patients. So the study was done on cell lines, and the enzymatic assay and other parameters were studied, and there was a difference. And so the difference is there, and the studies are being done. It is under publication. Once the publication is done, I think there would be some clarities in this. But as far as the observation goes, the difference will be there if we study the effect of Shastrokta Kashaya and the tablets also.
Sorry, I think you have observed one thing on an in vitro study.
Yes.
But then to generalize and say that there will be a difference is not in scientific temper. Okay. So while doing the in vitro study, what were the sort of things that you did with that formulation? You can't put it onto the cell. So you obviously did something to the formulation.
Yes.
So while that is a study, it's not rubbish. It has got meaning, but we cannot take that one experience and say that they are not equal. Okay. That's all I'm saying. I'm not saying yes or no to that, but that one study cannot say that it is different. That's all I'm trying to say.
It was about Rasavirya Vipaka, and for your information, that as there are receptors for the taste, now there has been a receptors for hot and cold. So Shitavirya and Ushnavirya, these are also received. These are also sensitized by the cell. And that way, that is one of the form of development of Dravya Guna Shastra to give an impact on the effect of the medicines, that we can explain the pharmacology in the three terms. That is maybe by Rasavirya Vipaka. Secondly, by the secondary metabolites, the impact of secondary metabolites. That way we can develop the pharmacology. And the third is the taste receptor, where recently it has been developed a cold taste receptor and a hot taste receptor. Yeah. And this is the way that Shitavirya and Ushnavirya, that can be detected by cell lines also. That's a very important.
Sir, let me finish Dr. Padki's questions. There are two part of that. One is the Guduchi. The second one is the PKPD of the tablets. Regarding this Guduchi's toxicity, I just wanted to call upon the entire fraternity of Ayurveda to report this pharmacovigilance. Fortunately, there is an Ayurveda Pharmacovigilance Center cell has been opened in Jamnagar. Okay. How many of us have reported to that? No, because we believe whatever we give, we will not have. One question that I have faced, I'll tell you, the Ministry of Health in Mongolia. Okay. Recently, the last week I was supposed to be there, but they are busy somewhere. So the next time I'll go. They wanted this answer. What is that? First question was, can a medicine be prepared out of herbs? They never heard of that, it seems. The new team. I said that is, the most blunderous thing that one can ever think if you are scientific so enough. Because the word drug itself, by the English word, which is French word says, drogue, dry herb. So drug is made from herb only. So then what is the big thing? The second one is, if it is a drug, second part of that, how can it be free from toxicity? That is the second question. Do you have a data? So this is what is very, very important, that any kind of these, the toxicity that we encounter, irrespective of, because there is the big format wherein what is causality has to be established in that pharmacovigilance report. That has to be done every time that we do. When we build up this big data, I'm sure that the whole world will stop reporting things like this. And one of them recently, Professor Malaji shared that one lady who got lead, that was lead, high level of lead, more than 80 nanograms per ml, which is more than 8 times than the normal ones. And that the report of the lead toxicity in the world, the environmental lead toxicity is more than you consume Nagabhasma, for example. Okay. But that kind of a difference that is not established because we don't have a big data recorded. Thankfully, now Ayush Ministry has opened. So let us all report whatever the slightest we feel there is something wrong with this our product, some effect that is seen. What you call ADE, there are differences there that we need to learn that pharmacovigilance, that is one. The second one is the PKPD, the WHO has defined for herbal medicine, PKPD is not necessary for the reason that these products have multi-compounds. For PKPD, there should be one compound. So one compound, and as you know very well, in a herb has got an umpty number. See, neem, out of 40 years, we have landed up today in 157th compound known. How many are there in neem? God only knows, or God may not be knowing also. Okay. So in that situation, it is impossible because it's so complex that herbal medicine they have waived off. So PKPD don't bring. That's why in the CTD, PKPD is a very important thing. Every minister asks. So WHO's document closes their mouth.
Sir, I have a question. Since many decades, Ayurveda industry is really taking the advantage of contemporary industrial things in the pharmaceutical forms and many ways. And what about the preservatives? As you already know, we'll be using sodium benzoate. We may be using many adhesives, binding agents, colorings in the tablets. Why Ayurveda industry not really invested any time, money to find our own preservatives? Of course, we have some classical preservatives like honey, many things, natural things. But do our industry really invested money and time for identifying our own things? Why it is so?
Good. As you know, the sugar is the best preservative. Okay. 63.5% if it contains, it is the best preservative. That is why our avalehas or syrups or whatnot else. Okay. And what is the situation today? Even 1% of sugar, gone case. So you need what? SF, sugar free. Okay. Because of whatever the reason is. Sugar has been made the culprit in diabetes. If it is so, Navagudan, what Prameha, yes, which is described in Ayurveda. So based on the Gura is nothing but sugar only. Okay, not a big deal. But what has happened? Sugar is in that level, 63.5. If you reduce to 60, gone case. Okay, that is number one. Number two, this amla paste in Chavaprash is preserved because amla is fried in ghee. Ghee is a preservative. Now, looking at that, the whole world was crying, so jumping, that amla is the richest source of vitamin C. Can you ever imagine the highest thermo level vitamin C is still present when you fry it in a ghee? Gone case. So it is not so. Later stages, as you can find that the emlicanins, the tannins, polyphenols A and B were responsible for its Vayasthapanam, anti-aging, not the vitamin C. Vitamin C was 1923. In the CFTRI in Mysore has established even why Charaka said Kara Amalakam Adhyayam. Kara Prachitiyam Amalakam Adhyayam Vyakyasamaha. When he said the second of the Rasayana Pada, okay, is Kara Prachitiyam. You have to go climb the tree, pluck and take it. Then only Rasayana. The CFTR has proved that the amalaki which is plucked and kept in refrigerator and room temperature, every 24 hours, it loses 10% of its ascorbic acid. In 7 days, there is nothing. So that's very sensitive. Now, coming to these preservatives, whatever that you described, believe me, these preservatives have been known very well, established very well, okay, preventing this bacteria, fungus, and these two mainly, and aerobic and anaerobic organisms. This is very important. Otherwise, the whole preparation gets fermented. These preservatives today is established in the pharmaceutical science, the limits for their use for their effective preservation. Anybody who is adding simply like that, dalo, sodium benzoate, and then make it so acidic, many of the kashayas are so acidic. That is very unfortunate. That is very unfortunate.
Oh, sorry to interrupt, sir.
And many times, last part is that you will be very surprised. These preservatives are not stable. They are actually degraded or denatured over a period of time. That's the biggest challenge in liquids and having high water activity products. That's very, very, very sensitive. So with all these, which is very well known, this what's that? Propyl paraben and parabens. Parabens even today in pharmaceuticals are non-toxic and the best preservative ever. But unfortunately, the whole world, particularly the natural world out in the West, says paraben to ban. Very unfortunate.
Yes, so I have one important question. While practicing also we face this sort of question, how we can make Ayurveda formulation cost effective? Because nowadays we are seeing high market prices of so many Ayurveda products. So Bhupesh Patel sir can.
First of all, I am saying the practitioners are prescribing multiple medicine. Say for 7, 8, 9 yogas are there, and for prescribing this multiple medicine, they have no exact idea which will act exactly. So first thing is to avoid multiple this prescription. And for that, we have to select, and we have the idea for selectivity, which particular drug is drug of choice and how it will act. So I think this is the only thing, because everywhere when I see multiple drugs are prescribed. So we have to stick on first, we have to identify which particular drug can be drug of choice by seeing the dosha, dushya, and particular whether it is for santarpana, whether it is for aptarpana, and it will act at which particular rog marga, which dhatu or dushya. And if we see detail all these things, we can cut down the number of yoga. So in my practice, I write 2 or 3 yoga only, and whereas this is the only way. Otherwise, we can't.
So you have given one important point, that is selection of the particular medicine. What is your point, sir? That how we can make Ayurveda cost effective?
Why to make cost effective? No need. Why we are compromising with the quality of the medicine, making it cost effective? That is most important. Second thing, what sir told, that targeted medicines should be used, no extra prescriptions. Whatever we see with the prescriptions in today's era, that people write hundreds of medicines. Already one formulation has number of ingredients into it, which has thousands of phytochemicals into it. We don't know the mode of action. And that's why we should not be behind cost effectiveness, compromising the cost. That's very important. Second, I want to answer regarding the preservative. So regarding preservative, when we think about preservative, when it has come from modern medicine, and modern medicines, preservatives, they restrict bacterial growth, fungal growth, or they restrict deterioration, not affecting the effectiveness or efficacy of the medicine. We have not at all studied the effectivity, how the effectivity has lost during the days by adding preservatives. We have not at all seen it. First thing. And second thing, now we are into the stage of giving 50 cosmetic formulations to an international brand, where we are not using any preservatives, artificial preservatives. We are all using natural preservatives. Natural preservatives can be used effectively, which can preserve the cosmetic salt drugs for at least 6 months. So that can be done, but efficacy should not be compromised, and cost should not be compromised against the efficacy. That's all. Thank you.
So we got the both 2 important points. So we'll come to you. Third point, sir, from you.
Hello.
How we can make Ayurveda cost effective?
Yeah, first only, he told nicely, he put up nicely that idea that it should start from the prescription. First point. Second point, of course, that's why the Rasa medicine came into being. When you are using Rasa medicine, effectively you can minimize the cost.
So second point.
And also, the Rasa mercury is a very good preservative. When our tetanus toxoid is preserved by mercurial, thimerosal. Thimerosal is nothing but thiomercury salicylic. So that's why the mercurial, so many mercurial came in Kajali form, in Parpadi form, and Sindura form, many forms. All these forms, we can reduce, remarkably, we can reduce the cost of a prescription. And we'll get very good results too. And the shelf life is more.
Yes, so important point is actually provided by you. Thank you, sir. So it's now over to you.
So here we have to bridge up something because we are living in a globalized world where there will be multi-companies, multinational companies are investing so much amount, and we are forced to take this. So in clinical setup, we have to develop a balanced thing. So some of the medicines that is uniquely produced by the companies, we have to purchase it because 70 ingredients, 90 ingredients and bulk amount is there, we have the. Another thing we have to keep is we have an independence. A Vaidya has an independence to select the medicines from their surroundings. So at least if you start to do 30 or 40% of drugs from your surroundings with a sustainable collection and utilization, then the whole thing will be changed. The economy will be balanced, our clinical practice, because we are living in a country like India where there will be a floating population where the economy is very important. Because the medicines cost, if it is increased, then it's not viable to practice. So which oil is necessary? Sometimes there will be only Murchida Tiladaila is essential for such a type of condition. There we will go for Mahaprasarana Tailam or something like that. It will be a difficult situation. So such type of things we have to take in considerations, and my humble request to all the young students are that at least the essential medicines, Agraushadhas, if you look the Agraushadhas, everything is in your surroundings. It's not some other things that will be coming from the very rare things. So if something is not there, you start to cultivate some of the 2 or 3 species and keep it like that. Such type of drugs which is having a multiple pharmacological activity in your clinic, you can add with some other drugs and we can practice. So that makes the sense of Ayurveda into another level, and that is the beauty of Ayurveda. There you can have a independence to practice in a different level.
Yeah, just to add on to the entire inputs, cost effective means 3 aspects. First of all, it has to be effective, and cost does not mean that we are selling at a lesser price. We can sell at an equivalent or a higher price also to any pathy in this world, subject to our treatment is getting effective as far as the patient is concerned. They are ready to pay anything if they are getting effect, because they are already suffering from so many issues. If you are charging 10 times to whatever they are paying to the existing option, they are ready to pay unless or until you are giving them the relief to the problem which they are suffering. Point number 1. So I'm not asking you to charge unethical, but I agree with the points I mentioned. If your Swarn yogas are expensive, but if after giving that, if MDR case, multi-drug resistance case, the entire antibiotic profile is resistance, and then the culture, the infection is rising, and if you are giving Jamangal Ras and you are getting the reduction in the culture, so that means the entire antibiotic profile is not working, your Jamangal Ras is working, patients are ready to pay 200 rupees also for a tablet, period. So do not focus on always cost effectiveness, focus on the efficacy of the treatment, point number 1. Second, always directly, let me complete, at least I will take 3 minutes because I have traveled straight away from Delhi for this event. So please allow me 3 minutes now. I keep quiet for the entire session. So let me complete, please. The second aspect is, as far as the dealing of a Vaidya is concerned, it should be directly with the brands. The maximum you deal and interact directly with the brands, the more you get opportunity to learn about their research, the better discounts, the better deals you will get for your clinic, which you can pass on to the patients. Third aspect, which correctly sir mentioned, that look around for the surrounding herbs. Fourth aspect, you can do your own white labeling, your own branding as well, in which you can bulk buy or maybe purchase a huge amount of medicines if your practice allows, and then can also you can save cost and pass on to the patients. And if you are at that level that you can have your own rashala, you can prepare your own medicines, why not? So there are many levels in which you can get involved, and definitely if you need any medicines from the companies like ABP and Himalaya, you have to get that at your clinic. But if you can prepare some medicines on your own, please do that. So point is, always ensure that all these permutation and combinations are applicable in your practice, and whatever price you are charging to the patient, it should make sense to them. If we are charging 500 rupees Swan Prashna every month, they are ready to pay unless or until their kids are seeing improvement, period. So anything which you are focused and doing with confidence and getting results as far as classical medicines are concerned, People are ready to pay unless or until you are showing them the results. That is the approach we need to take forward now. Otherwise, nobody will take us serious. And sometimes iPhone is selling at 80,000 or 90,000 because that has quality. So be iPhone in terms of any system of medicine rather than a Chinese phone. Thank you.
So thank you so much from. Sorry.
Yeah, I want to add to that also. I would like to, again, like he was saying, flip it on the other side, you know. I think Ayurveda is personalized medicine. It is natural, organic medicine. I think the very concept that it is to be cheap defeats the whole thing. In the world, anything that is organic, anything that is natural is the priciest. Vanilla natural costs 3,000 rupees, while synthetic vanilla costs only 50 rupees. So I think we are undervaluing our own science. We should flip it on the side. We are giving the most premium medicine, which is the safest, which is personalized. Unless we change our perspective onto that and give value to our products, it is definitely going to be the case that people value this as even scrap. I think that is one perspective. The other things, of course, the points that you mentioned, it has to be sensitive to we must look at, are we giving value for money? That is the question. It's not that it has to be cheap. If we are getting X amount of money, we should give X plus something in value to them. The value that they realize has got to be, there is something called a customer surplus. Customers should get a value higher than the value that they are paying out. I think that should be the perspective that we should look at, rather than saying, how can we be cheap? If you are cheap, very difficult for you to give high quality, is what I would like to add. Thank you.
This is from the perspective of the pharmacy, basically. From the perspective of the patients and the Vaidya, we have to really think that we will have to treat the patients of different kinds. If they can really afford to pay for this, we can just think about, or all whatever is expressed here is true. But if we have to treat patients who cannot afford to pay for this, it's not that they are devaluing it, but they cannot do it. They really want Ayurvedic care and cure, and they are not able to pay for it because they don't have money. Then what we need to tell them, this is what I used to tell my patients, that their participation in the healing process, that has to be enhanced. They should be ready to do that, because we are ready to tell them where these medicines are available, or we can even give the raw materials to them. They should be ready to make the medicines at home, and if that is possible from their side, the cost can be reduced to a great extent. But if they are expecting all the premium medicines in the great packages, and which is really looking very good, and still if the price has to be less, it is impossible. So if only the quality of the medicine is important, if they can participate in the healing process, then we can make the whole treatment cheaper.
The question is cost effectiveness. Cost effectiveness doesn't mean cheaper. That's very important. Effectiveness and the cost of that effect has to be paid for. And if you look at it, even from the Charaka time, the Rogi's Chatur Guna is Adyo Rogi first. Why didn't he say that poor Rogi or Bishak Vashya is the next one? Very important. It should be completely in faith with the physician. But he previously, Adyo Rogi should have. So why did he say this? It's very surprising that you look at it. So it cannot be cheaper. So cost effectiveness of that cost. Cost paid for that effect.
There are two things, like just last two observations. One is, we all are afraid of sugar as preservative. You know, one lakium, 1 teaspoon, how much calorie it has? Hardly 16 to 20 calories. If we properly control our diet, even for diabetics, nobody has not to tell sugar. If you properly do a calorie restriction, even with specific, so I'm playing on their part, even if we stick to our kalpanas, which are traditional, if Bishak Vashya Rogi is there, you can still perform. Second thing is misconception that when you mix many medicine, it becomes expensive. Our documentation we have been doing for last 15 years, it's rather opposite. You give a mixture of medicine. Now, the people who come to Ayurveda is not coming with one disease. It is in Vyadhi Shankarata Avastha. And it is very difficult. All the practitioner, do you have one patient with which you can only give Rasarayana and Adi Kashayam? Impossible. Impossible. That can be in text. So, what we have found out, when you make the precise combination of different dravyas, I'm doing a research on diabetes, what we found out, when we gave this complex formulation against one formulation, that HbA1c reduced in complex formulation by 12% within 3 months. And when we gave only one medicine, Amrita Churnam, it hardly reduced by 2%. Now, the decision is that it is more effective and what kind of situation you are dealing with. So if there is a complex situation, I think this is a intellectual freedom given in our Shastras. You don't have to worry if there is a yukti behind, whether you use one combination or hundreds of combinations. If you can do some prapti vigatana, if you can do properly treat disease, you do anything.
Yes, thank you so much. Both the sides actually, they have give a good insight about we have actually reached in depth to the topic, and many practitioners and many, like we are actually not aware while practicing about so many aspects. Like I have worked during COVID, that time I used Shaddar and Churna, and while talking with this panelist, I came to know Ativisha, instead of Ativisha, Musta is actually many a times is available. So I have till date, I was in my imagination that I'm using Ativisha. So there are so many things that we still need to learn, when we pass out from the college, actually, thereafter a real, the journey starts when you are into practice or when you are into manufacturing. There are so many challenges, and this platform is there to, you can evolve yourself. So we have contemporary plus classical side, both agreed on, so many parts where of the discussion. There are, debate as well, that they are not agreed at certain point, but definitely world is demanding Ayurveda. There are so many, like Biodiversity Act and everything has come up, and pharmacies are also, GMP is also there. So we have talked about everything. So now this is the time that we should conclude the session, because sir is also need to travel. So hope this is brainstorming and it gives you some insights.
Good evening, ladies and gentlemen. Namaste. A wonderful session on herbs and formulations was conducted. Both the sides were on fire, and I hope our delegates had a very outlook on both sides of the panel. All of their Purva Paksha, where they share their views in detail, will be available for the participants on the website of the event for watching anytime. We want to apologize for any kind of discomfort that you felt during this session in the name of good Tadvidya Sambhasha. Now, we would have a pleasure to honor each of the panelists for sharing with us their knowledge, opinions, and insights. Let's have a huge round of applause as I welcome Dr. Bhaswati Bhattacharya, a practicing integrative physician scientist, clinical assistant professor at Department of Medicine at Cornell Medical College, New York, to felicitate the panelists. Vaidya Rangesh Parmesh. Let's have a huge round of applause for Vaidya Rangesh Parmesh. Next we have Shri Ranjit Puranik sir. Vaidya Somit Kumar. Vaidya Bhupesh Patel, sir. Let's appreciate the insights which they provided us in this session. Vaidya Mukund Sabnis, sir. Vaidya Shashikumar Natural, sir. Vaidya Ajayan Sadanandan, sir. Vaidya Piyush Juneja, sir. Vaidya Ramesh Varier, sir. The round of applause continues for respected Vaidya PMS Raveendranath, sir. The claps continues for Dr. Jyoti Joshi, the session chair, for directing the session on the right track as required. A round of applause for Miss Anisha Nabi, who managed to keep the flow of session very smoothly. Indeed, a very remarkable session. Let's wait for a moment for a photograph. Thank you, everyone. There's an announcement. As I'm glad to announce that there is a session at 6:30 today where a Tadvidya Sambhasha on Ayurveda fundamentals led by M. Prasad sir and Kishore sir with joined by some other panelists would be there. So I would see you all at 6:30 today. Thank you, everyone.